In this video, we're going to look at how muscles actually move on the protein level in a process known as the power stroke. So myosin is known as a motor protein because it will take ATP and use that energy from ATP for movement. Now, initially in your muscles, we have myosin bound to actin. And the first thing that happens in the power stroke is that ATP will bind to myosin. And this binding event makes myosin release actin. Now, when ATP binds, myosin's active site will close around the ATP, and ATP will be hydrolyzed to ADP plus phosphate. And this hydrolyzation causes myosin to switch and actually move forward into its high energy conformation. Now, what happens after this switch is that myosin will now bind actin. It will bind actin again at a new place. So this orange is the original place myosin bound. Now it binds closer to the Z disc, and it will bind weakly to the actin. So after myosin binds, it will release the inorganic phosphate, but it'll still have ADP bound. And the affinity for actin will increase until now it's binding actin even stronger. Then once that inorganic phosphate is released, immediately what happens is the power stroke. The myosin head shifts back towards the C-terminal tail. That causes the thin filament to move backwards. And this can have a big movement of 100 angstroms towards the myosin towards the Z disc. And so what happens is that, like I said, your thin filament will move. And then after this power stroke, the ADP will release, and now we're back to where we started. So imagine that you have all these heads working in unison to move the actin filaments back and forth, right? And so that's how you get your muscle contractions. Now, we will only have muscle contractions in the presence of calcium. Or the power stroke will only happen with calcium. And that's because, if you remember from last video, we have tropomyosin and troponin on the actin. So this is blocking the myosin from binding F-actin. However, when you want to move your muscles, what actually happens is that nerve impulses will release calcium from the sarcoplasmic reticulum. And so the extracellular content of calcium will increase about 100-fold. Tropomyosin and troponin will bind this calcium, and this will cause a conformational change in these proteins, and the proteins will be released from actin. Now that the F-actin is cleared, myosin will grab actin, so it will have an affinity increase about 10,000-fold. But the calcium is only there for a very short amount of time. It is rapidly pumped back into the sarcoplasmic reticulum because you don't want your muscles to continuously contract. You want it to be controlled. And so when that calcium is gone, tropomyosin will change back to its original conformation. It will outcompete myosin, bind actin. Your myosin will come off actin, and your muscles will now relax. Now, actin and myosin just don't appear in your muscles, but they are in a lot of other tissues as well. Remember I said actin is the most abundant cytoplasmic protein. In fact, it's 5 to 10% of proteins in your cytoplasm. And in non-muscle cells, you get these microfilaments form. You get this fibers that forms from actin, which we call microfilaments. And microfilaments are F-actin again. And remember, F-actin has a barbed or plus end and a minus end. And globular or G-actin has a higher affinity for the plus end than the minus end. Now, G-actin will bind to both. It just binds more often at the plus end. And what happens when G-actin binds and becomes F-actin, the ATP in G-actin is hydrolyzed to ADP plus PI. Now, this ADP F-actin does not want to bind other actins. That's why affinity is lower at the minus end. Here at the minus end, you have ADP actin. At the plus end, you still have your ATP actin, which increases affinity. And you have this steady state happening where you have actin coming off at the minus end and adding at the plus end, so you don't have an overall change in length of the actin, but you are changing monomers. So if you follow this green actin, you can see that although the actin strand never changed length, it is shuffling through different actins. And this is called treadmilling when something happens like this, where your microfilament is constantly changing monomers but not changing size. Now these actin, F-actin microfilaments, they can exert a force on the plasma membrane, and this allows your cytoplasm and your membrane to stretch out in one direction, and that's how we can have movement in cells. That's what's being shown here. This actin is stretching out the membrane of this cell, causing this cell to stretch out. And that's it for this video. I will see you in the next one.